particular pleasure to announce a very special talk today. It is the first talk in the Coxeter Distinguished Lecture Series given by Natalia Komarova. She's professor at University of San Diego, but only very recently. So before she was a professor at UC Irvine for about 20 years, is this right? About 20 years, right? And so she's a well-known figure in the mathematical modeling of cancer. She doesn't need much of an introduction. However, I found a wonderful introduction here on Wikipedia. So you have a Wikipedia page. I don't know if you know this. So it says Natalia Komarova is a Russian-American applied mathematician whose research concerns the mathematical modeling of cancer, the evolution of language, gun control, pop music, and other complex systems. She's professor of mathematics and dean scholar at the University of California, San Diego. Please, Natalia.
Thank you so much for this nice introduction. I'm happy to be here at the Fields Institute. It's my third time, I think, and I always enjoy it. And I'm honored to give this lecture. So it's a series of 3 lectures. Today, I'll talk about stem cells. So there'll be some theoretical bits, and also I'll give you applications of this theory. And I'm one of those people who prepares 1,000 slides. And I was thinking about it, I can't help it. So let's see. So stem cells, adult stem cells, everybody has them. They are organ specific. They do not perform the biological function of the organ. They are protected in niches. They are immortal. And they serve as a reservoir of cells to replenish them as they do their job. So here's an example of a system, epithelial system. This is skin. So this is the surface of the skin. These are stem cells. You can't see them. When you go to the beach, get sunburned, these cells will die and get shed off. And then the tissue will be replenished from these stem cells. Stem cells give rise to more differentiated cells that actually perform the function. And this process of stem cell differentiation and them giving rise to more differentiated cells is very important. It's important for the maintenance of a healthy organ, and it's also important for cancer. So I'll give you a few examples of stem cell systems, lineages. So this one is what they call hematopoietic. This means blood. So if you look here, let's look here. So you will recognize some of these names from blood tests that you go for. And these are in your blood. But the stem cell that gives rise to all these different cells, from red blood cells and platelets to immune cells, the original cell is sitting in the bone marrow. Okay, we don't find this in the blood. So the stem cell is protected. And what's important for us is that different types of cells communicate with each other. Okay, they send signals. And I'll talk more about this. So maintenance of a constant cell size throughout an organ's life is called homeostasis. And it's very important for the healthy functioning of an organ. A mutant can refuse to do that. And that's the beginning of cancer. So some other systems that are relevant here is colon. That's an epithelial tissue. And we have neural system that contains stem cells and more differentiated cells. Olfactory, that's the sense of smell. So why is it so hard to maintain tissue constant size? You'd think it's so easy, right? But let's just think about it mathematically. Let's suppose we have some stem cells and we have differentiated cells. One of those performed its function and died, programmed cell death. So now there is a spot. What's going to happen? So the stem cell has to divide, and it faces some choices. There are 3 ways in which it can divide. There is an asymmetric division. It can give rise to another stem cell and a differentiated cell. It can self-renew, giving rise to 2 stem cells, or it can differentiate such that both daughter cells are differentiated cells. So if we denote by X the number of stem cells, X can go to X in the first place, X increases by 1 or decreases by 1. right? So let's look at the probabilities of these things. And now some very simple math here. If the probability of decrease for stem cells is greater than the probability of increasing, then the stem cells will disappear. There's no other way. If it's the other way around, they will expand. Not good either. So the only thing is that could work is that the two probabilities are equal to each other. But then, of course, we have a symmetric Markov walk, which results in either of these two outcomes. So this system doesn't work. It doesn't maintain what we need. So we need some sort of control that adjusts these probabilities such that the system is maintained. So this is what we call control. These probabilities are not constants, but they depend on what else is going on in the system. The problem is that nobody knows what goes on in the system. The information of how cells control each other is very difficult to collect. So mathematical modelers often assume things about these types of systems. So I go through a few pictures of other people assuming various control loops. So they draw arrows and they say, okay, this works. Let's analyze the system, right? We'll put different ways of control. So what I want to do here is I want to study all of these systems, right? I want to know what kinds of control networks are compatible with homeostasis. It's a very general problem. I don't want to assume a specific functional form or specific location of these arrows. I just want to kind of see what systems are stable and what happens. So we call this near equilibrium calculus of stem cells. So the concept of control, as we understand it, is pretty simple. So the divisions that a stem cell faces, so what decisions? To divide or not to divide, right? And once it decided to divide, it can differentiate or proliferate. This is the simplest, and I'll show you a more complicated system. So these are those probabilities, and we simply assume that the probabilities are controlled by the other cells that are at the moment in the system. So here's the simplest concept. A stem cell can decide to divide, the probability to divide, the rate of division rather, we call L, and it can be a function of X and Y, the number of stem cells and differentiated cells. And whether it decides to differentiate or not, that also depends on these things. And controls, we can draw them like this, as arrows. So these arrows, blunt arrows is negative control, pointed arrows are positive control. These are biological symbols. And so the way it's drawn right now, for example, this one, L is a function of X with a positive arrow. It means that the partial derivative of L with respect to X is positive, but it's controlled negatively from the differentiated cell. So that's negative. That's all it is. So control, so we assume that these rates are functions of what else is going on in the population. And how it depends on that, the sign of that is your control, right? Positive control, negative control. Or we can change that, right? And it changes the size. So we can write down ODEs. And we even saw some ODEs like this today, exactly these. We have the number of stem cells can proliferate or differentiate. These are differentiated cells that die. We can find the equilibrium. And remember, these things, L and P, are functions. So these are non-real, and we don't assume a specific functional form, right? So we assume that we can solve this, find a steady state, and figure out the stability properties. This will depend on functional forms. Or we can formulate a stochastic process. So we can do either of these. We can expand the system to include more compartments. For example, stem cells go to transit amplifying cells, to differentiated cells, right? And change the controls. So we derived kind of a general theory. And this theory is there to answer several questions, which can kind of characterize these systems of control. So the first question is, how do fluctuations of cells around their equilibrium depend on the types of control? You'd think that some types of control can be better than others, right? So without going into too many details, we were able to define exactly how the variance of the number of stem cells and the number of differentiated cells, how this variance depends on these partial derivatives on these controls, on their signs and on their values. Okay, so given the system of controls, I can tell you how much fluctuations the system will experience. Okay, for example, here in the 2-compartment system with 3 processes, we can find a closed form solution for the variance of stem and differentiated cells as a function of their controls. Another question that I wanted to mention that is interesting is what is the smallest number of control loops that is compatible with homeostasis? And it turns out that in this very small system that I started with, only 2 cell types, there are exactly 2 minimal control networks that each contains 2 loops. Okay, but if you now expand to 3 compartments that contain stem cells, transit amplifying cells, and differentiated cells, you have many more control loops. All of these have been identified as something that they're all topologically different from each other, and they all are compatible with stable solutions, so homeostasis. So the question is, can we make sense of this, and can we kind of figure out which one of these occur? So here's the first application of this theory, which came as a collaboration with David Axelrod from Rutgers a long time ago. So he heard me speaking about this theory, and he approached me by saying that I have data. So he has data from colonic crypts, 91 crypts, I think, which were taken from 94 patients. And they counted the number of cell stems, the number of transit amplifying cells, and the number of differentiated cells. So we have the means and the variances of these quantities. And he asked me, can you tell me what control loops are there? Right. So let's see. So we designed an algorithm of how to kind of narrow down the types of control loops that are compatible with this information. So we start with these, all possible ones. And then we get rid of the ones that have non-local control. So basically, we don't want this cell to control that. It's too far away, right? So, okay, that helps a little bit. Then we want to use the theory. So we know how the means and the variance depend on the controls. So given controls, I can calculate means and variances. Now it's kind of the inverse problem. I have the variances. What kind of controls is that compatible with? So that kind of got rid of some of these possibilities because it led to non-real solutions or negative solutions. Then we know that we have oscillatory injury recovery from data on other things. You remove cells and they come back, but they oscillate. So we only keep the loops that have imaginary eigenvalues. And then we have another piece of information, which is intra-crypt correlations that David Axelrod told me about it. So we only end up with 2. So out of, actually, it's even more complex. We later expanded it to 40 by including some other processes, and the answer is still the same. So these 2 are the ones that we hypothesize are there in place in colonic crypts. And I want to advertise this tool to you. It's very cheap. Instead of doing experiments and trying to calculate trying to measure which cell population controls which other, and it's not clear even how to do this, you can just do some mathematics. And that allows you to come up with a subset of possibilities, which can later be checked experimentally. So that led us to think about the principles of stem cells and their control. Perhaps economy and robustness are some of these. And another thing is perhaps the architecture of these loops is such that it minimizes the variance of the resulting solution. So the next example demonstrates why this may be the case. So we were thinking about a different epithelial tissue. It's the airway epithelium. This is the skin inside the nose of a mouse, which kind of is there to clean the air that the mouse breathes. And there are 3 types of cells there again. There are stem cells, and two types of differentiated cells. These are called secretory cells. They produce some goo that kind of helps retain some particles from the air to clear it up. And these are ciliated cells. They have little hairs that also serve the same purpose, to clean the air that an animal breathes. And so the question is, so we have this system again of three types of cells. It's a little bit more complex because the stem cells can produce the secretory cells, but also perhaps there's a direct way to produce ciliated cells. It's not a linear system like the previous one in the colon. And there could be other processes here, such as de-differentiation of these cells, and also both symmetric and asymmetric divisions. All of these have been hypothesized before. People studied the system a lot. We want to understand what goes on there and again apply our methodology to kind of figure out what the controls may be. So we wrote down all the possible reactions that can be there and just kind of paused, like what data are available. So there were three sources of data that we could find. The first one says the following. If we remove the ciliated cells experimentally, then pretty much nothing happens. So the system will recover later on, but very slowly, and so there's no immediate reaction to that. If, on the other hand, we remove both Y and Z, so remove both secretory and ciliated cells, the system recovers quickly. It's another paper. So each of these is a high-profile paper, a lot of experiments, very difficult to measure. The last one is remove X. So we remove the stem cells. And again, the system recovers quickly. Okay, so given these pieces of information, what kind of control loops are compatible with this behavior? So we can simulate this by assuming different sorts of loops. And it turns out that despite the wealth of biological information that was obtained through a lot of work, it kind of doesn't narrow anything down. We could assume all sorts of loops, and they all lead to the same kind of dynamics. So we're not able to exclude a lot of possibilities just based on this information. So it's very easy to find parameters that do exactly this. But then we did something else. So we looked at all the different control loops and found the one that minimizes the variance of the stem cells and differentiated cells. And it turns out that this specific system corresponds to the minimum variability of the steady state, right? So we were able to do that with our theory. So that's great. And then we found this paper. And this paper showed that it's exactly this kind of wiring that have been discovered in that system. So to me, it felt like discovering a planet from my desk, right? From just doing calculations on my desk and then seeing that some astronomers discovered the planet exactly where we thought it was. Because this specific system turns out to be the one that minimizes the variance. So that's why we think that we hypothesize that one of the principles of organization of stem cells should be the minimization of variance, okay, which is tied to this analysis that we created and could be thought of as one of the potential principles of organizations. So the next thing, of course, since this is a conference on cancer, is how do we break these networks, right? So far we have talked about healthy tissues and how they could be organized. Now let's see if there are any principles of how they can be modified by cancer. So we will start with a simple system that only has two compartments, but has a lot of processes. So we can, of course, expand this, but you will see even this is already sort of complicated. So we have stem cells and we have differentiated cells. And these are the processes that we could think of that could be there. Okay, so there's a self-renewal of stem cells, death of stem cells, differentiation of stem cells, asymmetric divisions of stem cells, self-renewal of differentiated cells, which could be possible, death of those cells, and even a back transformation, which is de-differentiation of these cells. So there are 7 processes, and we put them here in the set of ODEs. And remember that each of these rates, in principle, can be controlled by the 2 populations. And we don't know what it is, so we can assume various things. So there's a slight simplification of this. We can combine 2 pairs of these rates, which gives rise to only 5 parameters. So the ODEs now simplify. So these are all the processes that we have. So 5 controlled processes, 2 ODEs. Let's see what we can do. So what are the minimal loops that can maintain homeostasis in this system? Right? So it turns out that this is pretty easy to find and enumerate all of them. If the rate of de-differentiation is 0, so this is 0, we have 4 different loops. So for example, this one is where stem cells control their own rate of differentiation. Here, stem cells negatively control their own rate of self-renewal. So there are 4 of these. If we include de-differentiation, which could be there, we get 6 more. So altogether, we have 10 loops. So we can prove that in this system, 1 loop is enough, and there are 10 such loops. If some conditions are satisfied, we have homeostasis, so stable, positive solution. And so why are we doing this? What kind of mutations can break the system, right? So how can a mutant arise that can destroy the control? So there are 2 types of mutations that can be possible in this system. So here's an example of a loop. So these cells produce some signaling, and this signal is received by these cells, and it affects the rate of differentiation. So somebody sends the signal and somebody receives the signal. So 2 types of mutants are those that don't signal and those that don't respond. Okay, so with respect to the system, these are the 2 possibilities that we have. And by doing the analysis, it's easy to show that these are harmless. Mutants that don't participate in cell signaling cannot grow from low numbers. They are kind of in a system of ODEs, they're in neutral equilibrium. In a stochastic system, they're going to drift. So they're not advantageous mutants. On the other hand, these ones are the dangerous mutants. They are the ones that stop responding. And you can assume that they completely don't respond to the signal, or they can respond partially. So they kind of, there is a weaker response than there should be. Okay, so these are the ones that we will focus on. This is how we can incorporate them. These are normal cells. These are mutants. And the rates of mutants depend on X and Y with the coefficient. If the coefficient is 0, it doesn't depend on them. Otherwise, it just modifies it. It makes it as a weaker dependence. So how can we protect the system from invasion of such mutants? So we have these 10 different control loops. And the question is there a better and worse way to control, right? So 10 possibilities. If I am an architect, which one should I choose to protect the organ from these mutations? So I can cross out any of these loops, put the mutant in, and it turns out that they're all the same. They're all created equal. They're all vulnerable to the invasion of mutants that don't respond to the corresponding signal. So there's nothing for an architect to do here. But also, here's a simulation that shows that you put the mutant in and it completely replaces the wild type cells. And so it's truly advantageous mutant. Now, what can one do? And a design solution, of course, comes to mind is redundancy. Let's suppose that we add not a single loop, but 2 loops. We know that for stability, 1 is enough. But let's just use 2. In this case, if one of the loops has been broken, maybe the other one will help, right? So let's consider all the possibilities of combining two loops. There are 45 such combinations, but if we assume that a mutant breaks one of them, there are 90 combinations. So we can classify them by the behavior of the system once a mutant has been introduced, right? So I will talk about partial loss of response. So here, this table is organized like this. So these are all the control loops that can be eliminated by mutation, and these are the ones that are intact. So for example, here we have S controlled by X, and say here R is controlled by Y, and this one is eliminated, right? And the different colors, wait, sorry. So there's a color code here. So of course, red is not good. So red color means that when a mutant arises, then both stem cells and differentiated cells will increase, and they will go to infinity if they can even go to infinity under certain circumstances. So these are bad architectures because they're very vulnerable to the invasion of mutants. But there are some other colors which indicate a better arrangement. So green, for example, means that one of the populations can even decrease as a result of a mutation breaking one of the loops. And I show you two simulations that correspond to such great organization of control loops. Here, we control differentiations with the differentiated cells, and self-renewals are controlled negatively. Okay, and we break one of these loops, and the mutants arise, but then they reach a steady state that is actually lower. The stem cell state is lower in the new system compared to the old system. So this is very counterintuitive. You would expect that a mutant that breaks a control loop will always lead to an increase in the population, but this shows that there are examples where the loops are cleverly arranged such that the mutant actually cannot destroy this. In fact, it can improve the situation, or at least doesn't lead to any disaster. Okay, so that tells us that there are redundant control systems that are less vulnerable to mutations. And remember, in this very simple system, we had a set of 90 different combinations. So this reminds us of a different theory that many of you may know. This is the theory of Urielon, which is a theory of genetic networks. So they identified many gene regulatory networks, neural circuits, protein interaction networks. Many of them are possible, and only a small number of those networks actually occur in nature. So that was a big discovery. The idea here is that maybe in the context of stem cell regulation, something similar takes place. There are all these different possibilities, all these different ways in which cell populations can control each other, but only some of them may be advantageous for an organ. Those who minimize the variance in the healthy state, and those that minimize the harm that is induced by mutants if such mutants arrive. So that's kind of the end of this part of the talk. And now I'm going to switch gears a little bit and talk about something that is more practical, a system with some data that we recently worked on. So this is our attempt to apply this theory to a very concrete example of a hematopoietic stem cell system. I already showed you this very complicated diagram of different cell types in the blood. Let's take the first 5. Okay, so this includes the so-called long-term stem cells, short-term stem cells, MPPs, and these 2 branches. So remember, there are 2 very different branches of cells that are in the blood. These are red blood cells, platelets, granulocytes, and so on, and these are immune system cells. So the branching happens at this stage from the MPPs So we're going to keep both and cut the system there. So I'm going to take this same thing. So these are long-term, short-term MPPs, and the two types of more differentiated cells. Okay, and this is a paper that just came out in PNAS last week. So very new work. We wanted to take a very close look at this particular system and try to parameterize it because the data are available. So we wrote down a system of equations. It's exactly it's a very similar system to what I described before. There are proliferations and differentiation divisions of these compartments. And there are many parameters. So there's a parameter R, which is the rate of divisions. There's a parameter P for each compartment, which is the probability of self-renewal. And there are two parameters D, which is kind of like the rate at which these things go on to divide further. It's not death, it's just the further differentiations. So how can we parameterize this? So we use this paper, a great, amazing paper. I think it's extremely clever. That came out in Nature some years ago, actually. And it's about the neutral label propagation in mice. So what these people did, they inserted a certain substance in the long-term hematopoietic stem cell pool. So only these cells were, say, colored with some color. And apart from the color, these cells are completely neutral. They don't do anything different from any other cells. So they inserted this substance and analyzed the dynamics of this substance in the 34 weeks that followed. So let's think about it. We put it here, and there is an influx of these stem cells to the next compartment. So whatever fraction is colored, they're going to color these cells, right? And then it's going to spill over to the next and to the next. And then somehow it's going to go up and stabilize at the same percentage as it was here, right? This is pretty clear because these stem cells give rise to everything else. So the steady state will eventually be reached everywhere, which mimics the percentage of cells here. Okay, and these are the data. Of course, it's noisy, but we can see how this label propagates in time in these 4 downstream compartments. Okay, using that, we can take our system of ODEs and reformulate it very simply for the percentage of this label and how it spills over to all the compartments. So F is the fraction of this label that we see in each of the compartments. It's a linear system. And we also had an additional source of data from the same paper. There are actually a couple of papers, which tells us the size of the compartments. We don't even need the actual size, we just need the ratios of compartment sizes. And it turns out that this is enough information to parameterize the system completely. So we were able to find all the parameters. These are the fits. For the bars are this same data, and the fits are shown. And so these are the parameters and the numerical values. And it turns out that the numerical value is important. So just bear with me. So now we know the exact system of equations for this system. What can we say about it? What are some of the features that may be important here? So what we wanted to do is to study evolutionary dynamics of the system, because this is healthy. Now let's suppose that there is a mutant. What is going to happen to the mutant in this setting, since we know all the parameters, right? So this is a question that we asked. So now, what do we assume about a mutant? So we assume that, so these are, this is a system of equations for the number of healthy cells in each of the 5 compartments. They are called Xi. Yi are mutants, okay, in each of the compartments. And the mutants have only one difference compared to these. They have a modified rate of self-renewal. This is the probability with which a mutant divides and create more mutants in the same compartment. So this is kind of the most dangerous thing that can happen. A mutant that has an increased rate of self-renewal is usually a cancerous mutant, right? So we assume that there is a fitness advantage of mutants that expresses itself in these parameters. Okay, what is the fate of an advantageous mutant that we put in different compartments? So let's first put an advantageous mutant in this first most basic stem cell compartment. And it's kind of clear what's going to happen. There is more of it. It's stronger. It's advantageous. So the system is going to just completely be filled with such mutants. It's going to spread and replace everybody else. Everybody knows that. Okay, we are not the first people who wrote down this complete system. It's well known, right? So now, what happens if you put a mutant in the next compartment? Actually, this is less clear, or here, right? It's less clear what's going to happen. So we need to do the analysis. And this is just a linear stability analysis of a mutant-free solution. So we look at a solution that has no mutants, put a mutant, and see if that gives rise to, if this perturbation starts growing and leads to a different solution. So you can go through the steps. And so this has been done before, but what has not been noticed is there is a difference in the initial conditions. So if you put a mutant here, as I already said, it will grow. Any perturbation will grow as long as it has a slightly larger, any non-zero fitness advantage. But if you put it here or here, the conditions change. Actually, it's not so easy to, in this case, it's not as easy to perturb the mutant-free solution. And we have a barrier. In this case, if we put a mutant in this first compartment, the fitness advantage has to be bigger than the number. So given the parameter values that we know, that we calculated from the experiment, this is the fitness advantage that it takes. And if you put it here, you also need the fitness advantage. So let me just draw it here. So if you put any advantageous mutant here, it will invade. But here it takes at least 5.8% fitness advantage for it to be able to invade. And if you put it here, it's 20%, which is actually huge. In evolution, 1% advantage is huge. Because evolution is a long process, many divisions. And so, so 20% advantage is actually unheard of. And we are confident in these numbers because we parameterize the system. So it's not just, we have the analytical condition, of course, which is very simple. But here we were very surprised to see just how big this barrier is. What's the reason for this? Well, the reason is kind of obvious here. So if I put a mutant here, it outcompetes everybody in this compartment and then fills the rest of the system, right? It's advantageous. Now, if I put an advantageous mutant here, it self-renews more efficiently than anybody here, but the wild type cells receive help from the upstream compartment, right? So the mutant is competing, but this is not a fair competition. These mutants will receive an influx of normal cells. I'm sorry, the normal cells here will receive an influx from here. So it takes a certain amount of advantage to overcome that, and even more so here. Okay, these are the barriers, and there's a very simple analytical expression that you can calculate to find these barriers. And here's a simulation that shows this. Let's put a mutant that has a 2% advantage here. As this says, any advantage works, so these mutants are going to invade. They go up and they displace the wild type. If I put the same mutant here, it does nothing. It dies out because I need to have at least 5% advantage. And in order to overcome this barrier, I need a huge advantage. So we put a 25% advantage here, it works, right? Anything less than that will die out. Okay, there is a clever kind of optimization problem that nature solves here, which I wanted to emphasize. So there are two things that are going on here. So first of all, why do we have these compartments even? So one of the purposes is amplification of tissue. We start from a small number of stem cells, and we have to make more and more and more cells to eventually populate the whole system. So as we go from compartment to compartment, we need to experience a certain degree of amplification. So the amplification as a function of this parameter P, self-renewal probability, is an increasing function. As the self-renewal probability approaches 0.5, this is a log scale. This increases very much. So in order to have a lot of cells, to increase the amount of tissue, we want the self-renewal probability to be very close to half. On the other hand, we have this invasion barrier. And the closer we are to 0.5, the smaller is the barrier. So if we are very, very close to 0.5, the barrier goes down and any mutant can invade, right? So we face kind of a dilemma. I want to increase the tissue size, but I want to also have a decent barrier for the mutants to overcome. Otherwise, it's too easy, and the organ will have mutants, and that's the beginning of cancer. So the nature's solution to this optimization problem is these numbers, right? So these are you need a 6% advantage here, and you need a 20% advantage here, because this what is the probability to get a mutant here? What is the probability to get a mutant here? It's much larger here. This is a much bigger compartment. So if I'm more likely to acquire a mutant here, but then it has to face a 20% invasion barrier, right? So this is a clever organizational system that kind of tries to put down the probability of developing of cancer as much as possible. But nonetheless, as we all know, there is cancer. Leukemias are prevalent. Something happens, right? So how is it possible for the mutant to still invade and do their dirty job? There are a couple of hypotheses here. So let's suppose that we have a 2% of advantage of a mutant, and suppose this mutant appears in the second compartment, short-term stem cells. So it's not enough. Remember, you need 6%, so it goes down. Nothing happens. But now aging. Let's add the effect of aging. It has been documented that as an organ ages, the influx of cells from the upstream compartment decreases. And remember that this influx is what made the barrier, right? So if the amount of influx decreases, the barrier also goes down. So let's put that in. We're going to assume that the influx of cells goes down using realistic orders of magnitude. We don't have exact numbers here, but so, okay, so now we can see that the mutant doesn't disappear. It kind of even rises, but you know what? It takes too long. So these are all laboratory mice. They live 1,000 days. So everything stops here. And this is a log scale. So in 1,000 days of the mouse life, the mutants didn't die, but they are not dangerous, right? So this aging process is not enough. So something else goes on. So this is what our experimental collaborators are telling us. There is something else that is going on. For very many of these mutants, there is an ecological process that is called niche construction. So mutants create a certain degree of inflammation that affects the surrounding cells, but doesn't affect themselves. So this is a local kind of mechanism by which they give themselves a fitness advantage compared to the surrounding. So if we put that in the model together with aging, then the mutant can arise and win in the time frame that is compatible with the mouse life. But I just wanted to emphasize how it's kind of like an arms race, right? It's very difficult for a mutant to overcome all these barriers, but it manages by using all these different ways. And of course, evolution of the organism is such that it's trying to suppress that as much as possible and find ways to get rid of mutations by building these barriers. So this is what we have right now. And I wanted to thank my group and my collaborators and the support of grants. And we are hiring. So if you or someone you know are interested in postdoctoral position, please let me know. San Diego is beautiful, and I hope to have more junior colleagues there. Thank you.